Welcome to the 2023 Big Science Pitch. I'm so excited that you've decided to join us this evening. Thank you so much for making the time, and you are in for a lot of fun, as we all are. I'd like to begin by acknowledging the traditional owners of the land from where I'm joining you this evening. That is the Wurundjeri Woiwurrung people of the Kulin Nations. And I'd like to acknowledge Indigenous Australians who have been custodians of our lands and waterways for many, many thousands of years. I acknowledge and pay my respect to Aboriginal and Torres Strait Islander people as this country's first scientists and first storytellers, and I welcome all First Nations people joining us this evening. Hello, I'm Jen Martin. I'm an Associate Professor here at the University of Melbourne, and I founded and lead the Science Communication Teaching Program here because I think it's so important that scientists can communicate their work in engaging and accessible ways to a whole variety of different audiences. And we are going to hear some amazing science communication this evening. So this is our third annual Big Science Pitch. And I know that many of you have joined us before. Maybe this is your second or your third Big Science Pitch. Thank you for coming again. But for those of you who haven't been at a Big Science Pitch before, let me just briefly explain how this event works. So what you're about to enjoy is hearing from 6 of our leading early career researchers, and they are going to be pitching their work in just 3 minutes, and they are vying to share in a prize pool of $110,000 in research money that has been generously donated to the Native Australian Animals Trust. So each pitcher only has 3 minutes to wow you with their work, the problems that they're trying to solve, the questions that they are trying to answer. Each of them has been selected in a long and involved process. They've already been awarded $10,000 for being a finalist this evening. And then we've got an amazing judging panel who are going to decide on who wins first, second and third place. And you'll get to meet our judging panel soon. That's not all. We've got 2 other awards. The first is the Tim Winton Award. And this is a prize also of $10,000. Judged by beloved and multi-award winning Australian author Tim Winton, who is the patron of the Native Australian Animals Trust. Last, but of course not least, we also have the People's Choice Award, and that is the award that you get to decide upon. You're in charge of it. You'll get to cast your vote once you've heard from all of our pitches. So as you hear from each speaker tonight, please listen and think about who you would like to give your vote to. So please keep your eye on the chat. That's where we'll be sharing the link for the voting later on for the People's Choice Award, but there'll also be updates appearing there through the night. As well as voting for the People's Choice Award, there are a couple of other ways that we would love you to get involved. The first is that you get the opportunity to ask our pitchers questions. We'll have 5 or so minutes after each pitch, and I would love you to put your questions into the Q&A and let me know what it is that you would like to get answers to. If someone else has already asked a question that you would love to find out more about, please upvote that question to make sure that I'm going to be asking the most popular questions of each of our speakers. All right. It is now time to get started. I'm very excited and I hope you are too. So our first pitcher this evening is Dr. Laura Bibiana, I told her I'd get her name wrong, Dr. Laura Bibiana Ospina-Rosso. And Laura is here to tell us about her project exploring whether green Christmas beetles hold the key to keeping our cities cool. Sounds very inspiring, doesn't it? Something that you won't be able to Google about Laura is that she really enjoys watching wildlife documentaries, and she tells me that when she does, she really likes to imitate the animals' faces. So of course what I'm wondering is, how good is Laura at imitating a beetle's face? I'm sure we'll find that out soon, but for now, over to Laura for her pitch.
Would you wear black on a hot, sunny day? White may be better because it reflects light, getting rid of all that extra heat and making us feel effortlessly cool. Green Christmas beetles know it. Though less famous than their shiny relatives, these beetles have a secret. If you look really closely, you will notice that their delicate green cover is see-through, and underneath they wear a thick white shield that permanently reflects light. In our warming world, that can be trendy. In fact, if we covered just 2% of the Earth's surface in ultra-white paint, the planet would effectively stop heating up. That is like covering only 2 Australian states, Queensland and WA. Plus, super reflective paints already exist. So why are we not using them to cover our cities? Because they aren't up for the task yet. They lack resistance and durability. Now think about that green Christmas beetle. They are the hero of our story. Their white shield reflects an impressive 60% of sunlight and stays cool. Also, beetles have busy lives full of flying and sometimes falling. So I suspect this layer is also a very good armor. Strong, flexible, durable. How is this possible? We need to ask the beetle, and that's what I'm gonna do. First, I will study the white layer. If we zoom in with a fancy microscope, it looks lumpy and bumpy, full of wrinkles. I want to poke it till it cracks and stretch it till it breaks. By measuring its limits using museum samples, I'll examine its potential without harming living beetles. Second, I shall invite Australians on a quest to find our hero. If you see this lovely beetle, you can easily report its location online. I will go there and observe them in the wild to work out when the reflective layer is an advantage. If we can understand the green beetle's armor, we can apply this knowledge to build durable, reflective materials. So if you are excited about making our cities cooler and eco-friendly, vote for me, because this tiny hero may be able to teach us how to keep cool.
Thank you for your wonderful pitch, Laura. And I do notice that you're wearing white again. That seems very fitting, very cool. So I guess I'm really interested to understand, how did you work out that this white layer keeps them cool and maybe works as armour? Like, could it be doing other things as well?
Yeah, great question. Thank you so much. So our first approach was to take this white layer and we put it under a fake sun, so a simulator of the solar energy, and we realized that it reflects a lot of light, and we measure its temperature and it keeps cool. So, but we think that it may have other functions. So on one hand, that's what I want to test, but it could also work for other things like insulation. That's another question would be cool to address.
So potentially there's all sorts of things going on.
Yeah.
We've got a great question from the audience, which is, do we know what kind of temperature range we're talking about reducing, like from how hot to how cool? How effective might this layer be?
The current in the beetle.
Yeah, in the beetle, sorry.
Yeah, it's a big difference. So in their scale, the difference that we measure could be like only like 2 degrees, but in their scale, that means they get to be outside for longer.
Yeah.
They can eat more. They can find mates. All of that. So it actually is a big difference.
Yeah, I mean, 2 degrees is quite a big difference for everyone, right? But I guess if you're the size of a beetle, then that's a huge difference as well.
Yeah.
Another question has come in, and that is, do we know much about the composition of the layer? So you showed us that amazing image. We can see what it looks like, but what do we know about what it's actually made of?
That is part of the mystery.
Okay.
That is probably part of the kiting which is the material that makes up all the hard wings of the beetle, like the shell. But it just gets all wrinkly, and we wonder why that happens and how that happens. So it would be really cool to know how they actually make it that wrinkly. That's really new. So,
So, I mean, that's what we get excited about, right? Things that we just have no idea about that you're getting to explore. We've got a fantastic question here from Arn, and that is, have you thought of a solution maybe being something to do with glare? So we know that reflected sunlight can be very hard on our eyes. That's why we all like to wear sunglasses on really sunny days. Could there be something going on there for the beetle as well?
Oh, well, that's an interesting idea. I think it is possible, because if they wanted to camouflage, they wouldn't want to be like bringing out the attention with that glare. So maybe that could also be another function. That's why I want to go there and find them, see what they are doing. And if this question was also related to the application, that could also be, because our sunglasses have all these tiny bumps that also help to reduce glare. So it could also be.
So I feel like this amazing beetle that so many of us have memories of from childhoods, Christmas beetles being these really, really special creatures, could be holding the answers to all sorts of amazing questions.
Yeah, and we have, I guess, studied them a bit, but there is so much unknown about even their biology, where they are, what they eat. So this would be a great opportunity to just go and visit them, see what they do.
Oh, absolutely. And a question from Ali, do we know how these beetles are being affected by climate change?
That's a great question. For this particular species, we don't know. But in general, Christmas beetles have been affected by destroying their habitat, because that makes them obviously affects their food, they cannot find food. But also it makes them more exposed, and it's possible that changes the temperatures at which they are exposed, and radiation as well.
Yeah, okay, interesting. A question from Fraser for you, Laura, and that is, how would your understanding of this fascinating layer actually be applied at scale in our cities? It's obviously a big leap to go from something tiny on a beetle to imagining that it could, be part of the solution to warming cities.
Yes, that's so true. So some of these paints already exist, and what they do is they would create some bubbles in the paint that work similar to these wrinkles. But the cool thing would be to know if we can make them stretch more, or be more flexible, or be more durable in the beetle, and then we would be able to identify what feature is precisely what causes that. So then we can apply that in the paint.
Oh, Laura, it's such interesting work. Thank you so much for sharing your work with us. Thank you to all of you for your questions. I'm sorry we couldn't get to every question, but the ones that we did get to were just fantastic. So it is now time to move on to our second picture for the evening, who is Dr. Brendan Lanham. And Brendan is going to share with us what he thinks it is going to take to bring back our lost shellfish reefs. And Brendan isn't just into the science of shellfish, but also the science of beer. He tells me that he's a very avid home brewer. And of course, I'm picturing that beer and seafood probably make the perfect match. So thank you to Brendan. Over to you.
If I were to ask you what a shellfish is, I bet you're thinking about food. A delicious snack from the ocean, something we're looking forward to eating at Christmas time with our families. Something that definitely rings true for me. But did you know in the wild they form large reefs? I want you to picture the corals of the Great Barrier Reef. Reefs that have that beautiful, complex structure, crystal clear water, teeming with fish and other wildlife. A tropical paradise we all care deeply about. That's what we've lost, these beautiful reefs, but of shellfish. But why don't we care about this temperate paradise as much as we do about a tropical coral reef? You're not to blame. They're so far gone, they've been lost from our memory. We don't have something tangible to relate to. But I want you to care, because there's hope. We can bring these reefs back from the brink of extinction and restore them to their former glory. But restoration can be expensive, so we need to make sure we get it right. Make sure we're getting the best bang for our buck when we're bringing back these lost havens. But how do I think we can do that? By trying to replicate how these reefs once looked. We know that our shellfish reefs were once made up of oysters, mussels, and scallops. But what we don't know is how many of each species were present on the reef. Were they dominated by one species or another? Or were they there in equal numbers? Did they benefit from each other? Or were they in stiff competition, fighting for their right to eat a limited food supply? We just don't know. And that's what I plan to find out. I'm going to create small reefs in Port Phillip Bay that have different combinations of these species. And we can watch them grow from cute little baby shellfish through to temperamental teenagers and into thriving young adults. And we'll be able to see which combinations work best. Because if we can understand how to best make these species thrive together, we can have more efficient restoration. Which means we can bring back these reefs faster and over a larger area. So seeing these wonderful shellfish reefs surrounded by beautiful clear water, teeming with fish and other wildlife, and being able to see them on the Christmas table for years to come. If this is something that you would love to see, then throw me a vote, because I would love to see that as well.
Thanks so much, Brendan. I think now everyone has Christmas lunch in their minds. Everyone's picturing, what seafood am I going to have this year? But I'm interested to know, when did our shellfish reefs start disappearing? Is this something that happened a long time ago, and what led to their disappearance?
Well, it was almost directly related to the colonisation of this country. So initial harvesting methods were destructive harvest, so removing the shellfish so we could consume them, but also removing the shell base that they have underneath, so they grow on top of each other. The shell that's below them was used for the mortar in our early buildings. So over time, over the first sort of 50 years of colonisation, we started seeing the reefs decline very quickly, and the removal of those shells and that base substrate meant there was nowhere for the next generation to attach to, because they love to attach to something hard, mostly other shells of other shellfish. So that constant removal early on, they didn't have enough time to recover before, to replenish their stocks each year. And so, they constantly declined, and the removal of that base substrate meant that naturally they couldn't recover themselves, so they need a little help from us.
Yeah. Well, I'm glad that you're planning to help. That's very good to know. A question from Ol, and that is, where are you actually going to source your species from, your shellfish from? Are there still local deposits that you can go to, or how are you going to find them?
Yeah, that's a great question. So in Victoria, they're really heavily declined. So trying to find wild individuals to do this restoration is really difficult. So we work with a number of hatcheries in Victoria that they have some breeding adults themselves, and they grow them till they're quite small, so maybe 10, 15 millimetres, and then we purchase them from there, and then we can put them out into the wild.
At that size, do you consider them babies or teenagers? Are they temperamental?
They're not so temperamental just yet, so I'd still say they're babies at that stage.
Okay, well, that's good to know. Excellent. A question from Beck is, what's going to be the main benefit of finding out the right kind of balance and combination of species? Does this vary in different bays around the country? And how are you going to build them according to those kind of, combinations? Yeah,
so the variation is quite small depending on the species. So Victoria, the species we have here, are quite limited. It's not super relevant to, say, New South Wales, where their reefs are dominated by a single species.
Right.
And so the benefit of trying to understand the numbers of each species that we use is that we can try to tease apart some of the ecological mechanisms that are working between those species interactions, and try to maximise them. Often when you put two species together, they can benefit each other, and sometimes they'll compete a lot. And we don't really know what's going to happen in Port Phillip Bay, because we just don't have enough baseline data. And so we can try to really understand how to get them thriving together, and upscale that. So when we seed large-scale reefs, that we're not putting individuals together of different species that might actually be competing and we'll be working against ourselves.
Yeah. So much excitement ahead to see what actually happens when you put them together. Fraser is keen to understand more about the actual process of repopulating the shellfish in the ocean. Like, do you just kind of chuck them in there? How does it work?
Pretty much. So again, it depends on where you are. So again, this is where we see differences in different species. So if I was doing this in New South Wales, I would just put something hard for them to attach to, and there's a lot of larval supply. So there's really little babies really early on, and they'll attach to that and then tend to grow from there. Here in Port Phillip Bay, where we don't have that natural supply of babies, we do the seeding. So essentially what we do is we get those small individuals from the hatchery and we attach them to a hard substrate, usually like a scallop shell, and then they just get spread onto the reef. And so you put down a hard rocky base, cover that with the shells that have all the little baby shellfish on them, and then watch them grow from there. And then they can grow into adults and then reproduce themselves and then repopulate the rest of that hard substrate that's gone into the water.
So it's actually kind of simpler than we might imagine, yeah?
Yeah, it's not like overly complicated.
Yeah.
It's just trying to get them, once they're in the water, making sure that they're there, that they're surviving. It's the hardest part.
Yeah, of
and
course.
so this is where having those different species and trying to get them to work together can be really beneficial.
And so a good follow-on question then is, are these reefs susceptible to ocean warming and acidification? Like, you can get them there, but are they at risk of what's happening more broadly in our oceans?
Yeah, absolutely. So luckily, being a species that lives in cooler water, they're not under immediate danger, say like corals that are getting just decimated every year. So we've got a little bit of time to work that out. And the best way we're doing that is trying to breed oysters that are more temperature tolerant and that aren't so reliant on that. The processes of the acidification can slow down.
Yeah. Well, thank you, Brendan. I'm guessing everyone is now thinking, I'm hoping that not too many years away there'll be lovely local shellfish for me to enjoy for Christmas. So thank you, Brendan. That is all the questions that we have time for. Thanks again for all your questions. It's just great to have so many interesting things coming out from our audience. It's wonderful. But it is time to move on to hearing from our third pitcher this evening, who is Dr. Claire Taylor. And Claire has a really deep love for animals because she grew up in rural New South Wales on a sheep and cattle property. Not only does she have a farming background and love animals, but she's also an avid frisbee player and represented Victoria in the first ever Australian Masters tournament earlier this year. Sadly, we haven't got room in the studio for frisbee, but we can ask Claire about it later. But Claire is going to share her work with us on superb fairy wrens. Absolutely gorgeous birds, I'm sure you'll all agree. But how are they going to cope with our changing climate? So let's hear from Claire.
I'm really worried about what's going to happen to our superb fairy wrens this summer. In case you haven't seen the news, it's going to be another hot, dry El Nino year. Maybe you're worried about the increased bushfire risk or a massive electricity bill from cooling your home. But have you spared a thought for exactly how this will affect our native animals? We all know the changing climate is having a devastating impact on survival of many bird species. But what if it means they can't have as many babies in the first place? In hot, dry years, there are less insects for birds to eat, and water is scarce. This means mothers have less energy to invest in themselves and their babies. But just like on an aeroplane, where you must put your mask on first before helping your children, mothers must ensure their own survival before they can bring babies into the world. During my 8 years getting to know the secret lives of fairy wrens, who's having affairs with who and which babies survive to have families of their own, I've noticed that mothers usually produce less eggs at the start of the breeding season during early spring when the mornings are chilly, and they increase their clutch size as it becomes warmer until it's really hot at the end of summer when they produce less eggs again. Except in 2019, when most mothers didn't produce more eggs during the middle of the season. This was also our most recent El Nino year, when walking outside felt like walking into a full-body hairdryer. It was Australia's hottest and driest year on record, and unfortunately we could break that record again this year. I want to test if the number of eggs superb fairy wrens lay in each clutch is related to the weather. To be able to do this, I need to record the number of eggs superb fairy wrens lay in both favourable and harsh breeding conditions. Luckily, I know just the birds for the job. I spend each spring and summer finding the nest, like a treasure hunt, where the clues are the mother's nest building behaviour. Over the past few years, I've recorded clutch size in favourable conditions, but I need to record clutch size over multiple El Nino years, making this summer, now, a golden opportunity to do this research. If superb fairy wrens can't produce as many eggs this year, it could impact the maintenance of their population size, turning this species from familiar friends into rare occurrences, like those friends from high school you bump into once in a blue moon. It might also mean other species are at greater risk, as superb fairy wrens are common and have multiple clutches per year, but many species only get one attempt. That's why we urgently need to understand and quantify how climate affects animal populations, because El Nino years are predicted to intensify and last for longer, even across several years. If nature won't nurture our little fairy wrens, then we need to arm ourselves with the knowledge to help them when the time comes.
Thank you so much, Claire. They are just such beautiful birds, and I imagine everyone at home is saying, oh no, I need to know that they're safe this summer. So tell me, in a good year, when the weather is kind of ideal, how many eggs can a superb fairy wren lay per clutch and per year? Like, what are we talking about in terms of survival to the next season?
So usually fairy wren clutches go between 2 and 5 eggs, most commonly 3 and 4, but in a good year they can have 5. Okay. I've only seen this a couple of times, and last year we had one where all 5 babies survived, which was amazing. I got quite the surprise when I was pulling them out to band them and there was a 5th one. But yeah, in those dry years we really do see more 2 to 3 egg clutches, so it does make quite a difference, and they can have anywhere up to 8 breeding attempts per year. So a lot of fairy wren nests get eaten, up to 66%, so they keep trying and trying and trying until they have some success.
Who's eating them?
Yeah, everything loves eggs. They're delicious. So brown snakes, currawongs, kookaburras, rodents, heaps of things.
Okay.
Yeah.
Oh, all those poor eggs. Yeah. We've got a question from Robert, and that is it actually better that the wrens produce fewer young in hot years? So I think Robert's saying, are they actually kind of depleting their resources and ending up with all of the young dying if they produce too many young when the weather isn't good?
Yeah, so I guess having more eggs in your basket is kind of a better bet.
I see what you did there.
But it is important that they can feed those babies as well. So I understand the point you're making of perhaps they're reducing their clutch size so that they don't have starving babies, but we don't often see chicks or babies die in the nest. Okay. So I think, producing... It might be one strategy they're using, but I think it's also the limiting factor of not enough food to be able to produce those eggs.
Okay, interesting. Another question is, how many nests would you need to be able to find and look at and collect data about in order to draw some conclusions?
Yeah, so we follow about 30 to 40 breeding females each year, and as I was saying, they can have up to 8 nests. So we follow each of their breeding attempts. So we watch them to find their nests and then kind of follow them through to see what happens, whether they get eaten, whether they survive and fledge, where they build their next nest. So we would just collect as much data as we can. Yeah. And, obviously more data is better, but we just do as much as we can with those ones that we're following.
Yeah, of course. I mean, you can't, what do they say, a nest in the hand? Or there's sort of got to be some good saying there, right? You know where it is, you've got to get all the data. Yeah, exactly. A question from Tanya, and that is, are you looking at temperature variations or food availability and the effect that those things are having?
Yeah, that's a really great question. So is it the fact that it's the temperature making it really hot, so it's hard for them to thermoregulate and stay cool? Or is it the fact that it's really harsh and dry and there aren't as many insects available for them to eat? It could be both. both of those things would be great to look at, and I think it is important to consider all of these different aspects and, take them into account in our study as well.
Well, we've got another question, another thing to think about, and this is from Rachel. Do we know what sort of impact humidity has on clutch size? So would you expect to see the same decreases when it's hot and humid?
Yeah, that's a really interesting question. I mean, I guess when it's hot and humid, generally there's more plants growing and therefore there's usually more insects around. So hot and humid is actually quite plentiful conditions, whereas hot and dry is absolutely the opposite. So I think the precipitation or rainfall does play a really big role in how many resources are available. So that's a really good question. Yeah.
Look at all the budding scientists in the audience giving you all of these additional things that you need to be collecting data on. A more bigger scale question is just how long do these wrens live for, and how many years can a female successfully breed for?
Yeah, so they can breed the next breeding season after they're born.
Okay, wow.
Some of them hatch in January, so that's quite the feat to start breeding the next September or October. And I think the oldest female we have at the moment is about 6, but they can get to about 9, and they breed for that entire time. So it can be quite a while, but the average age is about 2 or 3, I think. Okay. Long enough for us to build a relationship with them and see how their lives develop.
Yeah, and get really, really connected with them.
Absolutely.
And so if you do find out that this hot weather is having really dire impacts, are there things that we can do to help these wrens be more successful in breeding?
Yeah, so that's the question, right? So what impact is it having? And I think it's one of those things where having more information is better. The more we know, the more we can think about if we need to put things in place and what that might look like. So do we need to be providing water when there's none available to be able to help them through, or what is it we can do? But before we can even think about that, we need to know what effect the weather is having on the wrens. Yeah.
Well, thank you so much, Claire. Clearly a very big year in the field, or months ahead in the field for you this summer. Thank you to everybody for your fantastic questions. And wow, what an amazing start to the night. Thank you so much to Laura and Brendan and Claire for sharing such incredible work with us. And of course, thank you for all of your excellent questions. I think now it's time for you to meet our esteemed judges. You already have a sense of what a hard job they had ahead of them this evening with the quality of our pictures. So our judges this evening are Professor Mick McCarthy, Tanya Ha, Christian Williams and Dr. Ellen Cottingham. So I'm going to begin by introducing you to Professor Mick McCarthy, who's the Deputy Dean of the Faculty of Science here at the University of Melbourne, and he's chairing our judging panel this evening, and then he will introduce you to the rest of his judging team. So Mick is a Professor of Quantitative Ecology, and his research focuses on ecological modelling, which means using mathematical and statistical methods to help us understand ecological systems and to develop approaches to improving environmental management and conservation. And he also teaches in a variety of areas related to ecology and environmental science. So Mick, thank you so much for heading up our judging panel this evening. You have quite a difficult task ahead.
Thanks, Jen, and thanks to our 3 opening pitchers, Laura, Brendan and Claire. It's been an incredibly impressive start to the evening, and yes, it's going to be a very difficult decision for us. They've set the bar really high for their colleagues. As you know, there's $110,000 up for grabs this evening, and that's thanks to the generous support of the Native Australian Animals Trust and also through the Robert Johansen and Anne Swan Fund. The Native Australian Animals Trust is a way for people to connect with Australia's nature, for those who are passionate about it and their environment, and supports the University of Melbourne's research, teaching and engagement activities. Australia has a unique and precious landscapes and flora and fauna, and some of the most beautiful flora and fauna in the world. Our animals require careful custodianship and unique approaches towards their conservation. It's our responsibility and and prerogative to protect and preserve our natural environment. It's for these reasons that we established the Native Australian Animals Trust. The Trust supports work on Australia's animals and their environment and has two broad purposes: to learn about Australia's animals and how to improve their conservation, and also to tap into the fundamental biology of these organisms to improve our own lives. Thanks to our generous donors, the Trust helps to fund our students and researchers to advance important environmental and ecological processes. It's now my pleasure to introduce our judging panel, and who will be responsible for deciding on the winners tonight. And you also get to have your own say through the People's Choice Awards. Voting for that will commence once we hear from the last three pictures. I have the honour of being one of the judges tonight, which is never an easy task, as each of the projects is worthy and important. I'm joined by three very talented experts who are dedicated to the preservation of our environment and native species, and it's my pleasure to introduce my fellow judges. First, we have Dr. Ellen Cottingham, who's a postdoctoral researcher in invasive species management from the School of Biosciences here at the University of Melbourne. Ellen knows a thing or two about what our panellists, our pitches are going through this evening. As last year's Big Science pitch, Ellen was awarded joint first place for her pitch titled Genetic Biocontrol of Smooth Newts, and we'll have the honour of hearing about Ellen's work a little later and what it means to win this competition and how it has helped her work. Next, we have Tanya, who's an award-winning Australian environmental campaigner, author, broadcaster, science journalist and sustainability researcher. Tanya is an environmental and science communication specialist. In addition to her media work, she also develops sustainable living and behavioural change programs. And finally, I welcome Christian Williams, a teacher from Melbourne, Australia. He has been recognised nationally and internationally for his teaching expertise. At Mentone Girls Grammar School, Christian manages the award-winning Enterprise Academy, a world leader in educating and inspiring and empowering young women for a better world. He is also the co-founder of one of Australia's top education startups, the STEM Circle. Thank you to our judges for taking the time to be here tonight. Our challenge is to assess the pitches on innovation, delivery and impact. After seeing the first three pitches in action, it's clear we've got a very difficult task ahead of us. I'll hand back over to Jen now and to introduce the remaining researchers.
Thank you so much, Mick and Ellen and Tanya and Christian. I'm so glad it's your job. I just get to introduce our pitches and have lovely conversations with them. I'm really glad it's you having to make the tough decisions and not me. But thank you to all of you for being in the studio with us. So I will remind you again, just make sure as you listen to our pitches that you are thinking about who you're going to vote for the People's Choice Award, and keep those fantastic questions coming, and you are able to upvote somebody else's question if you think they have already asked the perfect question. So it's time for me to welcome our next pitcher for this evening, who is Dr. Joshua Barrow, who's going to talk to us about fishways, something you may or may not have heard about. But Josh is interested in understanding whether fishways are a silver bullet, a wonderful solution for our native fish, or whether in fact they are a problem and an obstacle. And for all our Grand Designs fans out there, Josh tells me that he has a very big year planned for next year, working on his Grand Designs bingo card. Not only is he building a house in Gippsland, but he's also going to become a dad for the very first time, which is very exciting, Josh. So over to you for your pitch.
Many of you will have had a fish tank at one point in your lives. But have you ever wondered why some fish hide away in their little houses, while others will try and bite your finger off? Well, it might sound ridiculous, but just like Dory and Marlin, fish have different personalities. This might not mean much for the fish swimming around in your tank at home, but for the fish in our rivers, it can be hugely important. River systems are some of the most degraded on the planet. And one of the biggest reasons why is that humans have drastically changed these systems by building barriers like weirs and dams. These are very useful to us as we use them to get the water to grow the veggies that you eat for dinner every night. But the downside is that these barriers disconnect rivers so that fish can't move. Just imagine being a little fish trying to move through the river and coming across this. Movement is critical to our native fish because many species need to move to breed, explore, and survive. Managers and governments, they're trying to help. One way is by building structures called fishways. They're like a bypass on the road to avoid a traffic jam. Fishways come in different shapes, sizes, and designs, ranging from a bunch of boulders scattered across the river to carefully designed mazes that let fish move from below and above the barrier. Seems like a great outcome, right? Well, unfortunately, it's not quite that simple. Not all fish are actually able to swim through a fishway. One of the reasons why is because some fish are bold and some fish are shy. But how is a fish's personality relevant to a fishway? Well, it's likely that fish need to be bold enough or brave enough to successfully pass through a big, scary fishway. Think of the redfish here as the bold ones, easily moving across the barrier, and the yellow ones as the shy ones, getting stuck on one side. This not only limits the total number of fish that are moving through our rivers, but also bold fish are often more aggressive, they're more competitive, and they can be naive around predators, making them an easy target. I'll use a species called the common galaxid for this project, which is a native migratory fish that moves between rivers and estuaries to breed. I'll collect the galaxids from several fishways in Victoria that have different designs, and I'll assess how bold each fish is. I'll tag each fish with a tag that's similar to the one that's in your dog's ear, and put tag receivers above and below the fishway. I can then see which individual fish are able to move through and which ones can't. With the results of this project, I'll figure out if boldness is a major reason why fish are able to swim through the fishways, and if some designs are more accessible than others to a wider range of fish. Vote for me, and together we can improve how we design fishways so that we can get more fish moving, ensuring that they'll thrive for generations to come.
Josh, just awesome. And I'm picturing now that everyone at home is running over to their fish tank. And the obvious question, which Rachel has already asked, is how do you measure how bold a fish is? And can people do it with their fish at home?
I suppose so. I hadn't really thought about that. But yeah, it's quite an interesting one. We've got a master's student in our lab at the moment who's doing a few of these experiments. And essentially you go out and collect the fish, and then you construct a little bit of a maze, and you pop the fish in a little house on the side, and then you videotape the fish and see how long it takes for it to come out of the maze, whether it decides to stay within the maze, or whether it's happy to sort of investigate all of these new environments. And so you can get a bit of like a score for each individual fish's level of boldness, I suppose, from that.
And so do the shyest fish just refuse to leave their little house and say, I don't want to go in that maze, that looks scary?
Exactly right. Yeah, exactly right.
Oh, now I'm feeling sad for all the fish that don't want to go into your scary maze. A great question from Roger, and that is, do cormorants and other predators congregate around these fishways? Do they know it's a good place to find some shy fish who don't want to go in there?
Very possibly. I suspect that there are several different predators that do these sorts of things. I know that there are some examples of some larger predatory fish that actually just hang out at the top or at the bottom of a fishway, and then as soon as these fish pop out, they just can have themselves a very good snack. So I suspect cormorants would probably do the same thing because they're pretty switched on as well.
Yeah, I mean, as soon as you realise there's a whole lot of fish hanging around, why wouldn't you hang around there too?
Indeed.
A question from Fraser is, has there been any previous research on these fishways? Do we know much about, how fish are behaving around them?
There has been a fair bit of research on fishways. It's quite interesting. When they first built fishways in Australia, we really designed them based off European designs. Right. And that was quite interesting because a lot of European fishes, say you think of like trout, or Northern American, you think of trout and salmon and these ones, a lot of these fish are able to jump and swim sort of right at the top of the water column. And so when we designed these, our native fish really weren't able to move up and down the river at all. Mm-hmm. And so we decided to bring in all sorts of new designs that facilitated passage for our native fish. Mm-hmm. But generally what happens is there might be some sort of monitoring process that happens after you build a fishway, and you can say, yes, we're getting fish going through, and that's generally a tick-off, but there's not really too much that's looked into about that inter-individual variation in behaviours and things like that. Yeah. There have been some international experiments that have looked at sort of the different personalities and things like that, and mm-hmm. it seems to suggest that the bold fish quite do a lot better getting through the fishways than the shy ones. Which
kind of makes sense, but I'm guessing everyone at home has had the same response that I had when I first spoke with you about this, and that is, who knew fish had personalities? We tend to not really think about fish having different personalities. And Peter has asked, do different species have quite marked different personalities? Or I think you're talking about different personalities within a species, yeah?
Yes, I think both. So definitely there are some species that are generally just a lot more aggressive than others, mm-hmm. Whereas there are some that are generally quite secluded. And so we've done a bit of work up in the high country, for example, and we work on trout and we work on blackfish, and you'll generally find the blackfish hidden up underneath little corners on the river, and the trout are kind of in the mainstream, just sort of ready to go to attack anything that they want. Yeah.
All of those bold trout.
Yeah, indeed.
We've got a question, and that is that if we know that predators exploit fishways, are they like the fishways themselves, are they likely to be upsetting the natural balance? Do we need to find a way to minimise this kind of, way of congregating fish for predators?
I'm not really sure if it would be having that much of an influence with the predators eating fish at the top or bottom. I suspect that things like recreational fishing and other activities like that would probably be removing a lot more larger fish, but I wouldn't have thought that it would be something we'd need to really kind of target.
Yeah, okay. A question from, where did I just see one? A question from Vern, and this is a really interesting one. Do shy fish learn to become bolder by going through the maze? So, are you training fish to become more bold by testing whether they are bold or not, if you know what I mean?
I think that's a really good question. I suspect it might vary based on the species and based on the individual. It's very possible because a lot of fish can go through sort of all sorts of learning processes, and we generally would take several measurements per individual, so you can get these sort of repeated observations per individual. So I suspect it's possible that some fish would learn how to get better, but then it might also be possible that a bold one might come out, see a human, and then decide that it didn't want to be out there for much longer.
Big scary Josh, I'm going to go and swim in the other direction.
Well, actually, interestingly, there's some work that happens with hatchery fish, because we produce a lot of fish in hatcheries, which we then stock out into rivers and lakes and things like that. And generally speaking, these fish are quite bold because we feed them in the hatchery and then they all just try and attack as much food as they possibly can. but it means that they're also a little bit stupid when it comes to predators. And so there's some work that's been done to show these fish different predators and deliberately spook them so that they can learn how to be a little bit better around predators once they're put into the wild.
Yeah. Oh, look, Josh, it's just fascinating. Thanks so much for sharing your work with us. And now anyone who has rushed over to their fish tank at home is now got all of these brand new questions to ask about how smart are my fish and how bold are my fish. So thank you, Josh. That's fantastic. All right, we are already about to meet our fifth picture for this evening. It's going so quickly. Our next picture is Dr. Rachele Wilson, and she has all the buzz to share with us on conserving native stingless bees in Arnhem Land. Terrible pun, I know. But what you will be interested to hear is that despite working on native bees and wasps for 10 years, Rachele has never been stung by one. So that's a good thing to know. And at her home in southeast Queensland, Rachele has 12 beehives and 8 bee hotels. I'm imagining most people have a visual of what a bee hotel is. And they're all occupied by different bee and wasp species. And in case you were wondering, Rachele's very favourite species are the masked bees. So let's hear from Rachele talking about stingless bees in Arnhem Land.
For most people, if you want honey, you buy it from the shops. But for many First Nations peoples, they go to the bush. And I'm not talking about honeybee honey. I'm talking about Australian stingless bee honey. The Yolngu First Nation have been collecting stingless bee honey, or sugarbag, every dry season for thousands of years. They go out into the bush, look for the glistening wings of bees flying back to the nest, and listen for the droning of bees inside. But the past few years, this has become increasingly difficult because they're not seeing these streams of flying bees anymore, and they're not seeing many flowering eucalypts, which is probably an important food source. Now, stingless bees only live in warm, tropical climates, so if their populations are declining, there's a chance it could be due to climate change, since we know it's hitting the tropics first, with heat waves lasting longer. When temperatures change too much, this can really throw off the time that plants flower. So in the dry season, when it finally stops raining and the bees can fly again, what happens when their usual food sources aren't there? As a molecular ecologist, I find out which plants are food sources for bees by tracing the DNA from the pollens they carry back to the plants that grow in the region. But I have to find the bees first. Now, for most people, finding stingless bees is really challenging. They're very small and very quiet. But for Yolngu, it's a piece of cake. This year, I surveyed bees with some amazing Yolngu rangers in East Arnhem Land. Stalking flowers with our nets, we found over 20 different species of bees foraging on shrubs. Next year, I want to go back and find stingless bee nests in the trees. If we can find enough nests and take pollen from them regularly, I can find out which plants bees have been saving food from throughout the year. And I can look at the DNA of the bees themselves to see how related they are to each other, which tells us if the population is strong or dying out. A decline in stingless bees could particularly hurt Yolngu because for them, bees are a part of the very fabric of their culture. Like the people themselves, all stingless bee species belong to Dua or Yirija, the moieties that bring balance to everything in East Arnhem Land. This is a modern problem that requires collaborative solutions, and it's not possible without Yolngu knowledge. I need this funding to help Yolngu find out what's happening to their bees, to maintain their cultural practices around honey, and to conserve the bees and plants themselves. Thank you.
Rachele, thank you for helping us to understand a bit more about the fact that when we talk about bees in Australia, it's not just thinking about the standard honeybees. But I'm really interested to know, what's the actual process for Indigenous people to harvest this honey? I mean, it's very different to what we think of, a beekeeper in their suit going to an artificial hive. How do they do it?
Yeah, that's it was something I only just discovered very recently. So last year I went out there to North East Arnhem Land, and it differs depending on where you go and who you talk to. But for the people that I'm working with, there are certain people who have this knowledge, and it tends to be women, but sometimes men. And generally what they'll do is they'll look for these bioindicators to suggest that it's a good time to go out and get what they call guku. It's the local term for sugarbag bee honey. And what they'll do is they'll look for these blooms, mass blooms of eucalypts or corymbias, and then they'll see lots of stingless bees coming and going from those plants and follow the glistening of their wings back to looking at a certain angle from the bees to the sun. Follow those bees back to the nests in the trees. And then once they locate those nests, today you can use a tomahawk or an axe or a chainsaw or something, but obviously in the past they would have used some very different tools, and it would have taken a lot longer. But you have to chop down that tree, and then there's a very specific way of opening up the log very carefully to find exactly the part of the nest that you're looking for in order to get the honey, so you don't just destroy the whole thing.
So we're talking really important Indigenous knowledge here.
Yeah.
And you said the nests are really hard to spot, very different to what we think of when we hear, a hive. Have you got to the point that you can spot one of these nests, or is that still a skill being developed?
It's still being developed for me. I'm a little bit vertically challenged as well, so that doesn't help.
I know the feeling, it's okay.
And my eyesight isn't that great, but when they have what they call the long nose, so for stingless beekeepers we know this as the entrance tunnel. For certain species that have this long propolis entrance tunnel, it's like made out of tree resins and wax, they're a little bit easier to see because you see this weird thing sticking out of a tree and like, oh, what's that? But for ones that don't do that, it's really impossible. It's quite hard. And because it's such an untouched landscape, there's a lot of trees, it's a high density forest, then it's like looking for a needle in a haystack.
Yeah, I mean, what a skill to develop, though. I'm just imagining putting that on your CV one day. I know how to spot these incredible nests. And are stingless bees, this is a question from Juliet, are stingless bees the primary or the only pollinators for some native Australian plants?
Well, actually, we've only got about 11 stingless bee species in Australia, possibly 12. We're still figuring that out. And the rest, so we think we've got about 2,000 species of bees in Australia. So the rest of them are not stingless. They sting, and they're solitary species. So they're actually much more likely to be specialist pollinators of certain plants because they range from like 2 millimetres in size all the way up to more than about 2 centimetres. So yeah, there are certain bees that are specialise on particular plants in Australia. Yeah, but they're generally not the stingless bees. Stingless bees are what we call generalists. They're much like honeybees.
Okay. I've got a question from Jeff here, and that is wanting to understand more about and I'm not going to pronounce them correctly, the dua and the yiricha, is that right?
Dua and yiricha.
I'm so glad you pronounced them for me, thank you. Can you tell us more about what those two kinds are?
Right, so it means different things. So it's a way of describing which moiety you as a person belong to. And it can also be a way of describing what moiety different plants and animals belong to. So there are 2 main stingless bee species in East Arnhem Land, and these are classified as either dua or yiricha. But then within a nest, you can also have there might be a dua bee, but then they would call the eggs of that bee yiricha, because it's kind of like their equivalent of like yin and yang, hence why I said balance. So it's a way for people to understand, where they fit within society, but also what their obligations are to the other parts of society and to the landscape as well. Yeah.
Thank you so much for sharing that with us. It's something that I think everyone probably has this sense that they would love to understand more about. Jane is interested to know simply how many species of bees are there in Arnhem Land? Do we know that?
We actually don't know for Arnhem Land. So we think for Northern Territory there's probably about 200, so about 10% of the whole country. And there actually aren't many records of bees that have been collected in East Arnhem Land. So I've actually got a master's student working on that right now, and I suspect that a lot of the bees that we've been collecting in this past year and a half are going to contribute to those records. There are certainly things we're finding there that we didn't know were going to be there. So yeah, it's a bit unknown, and like To date, I think we've only got about 1,600 described species in Australia. That estimate of 2,000 is just based on the idea that we've probably got a lot that we haven't discovered yet and that haven't been named that are still sitting in museums. So, I mean, that's just for the whole country, and they tend to be in more urban areas that these studies are done. So, I think for East Arnhem Land, there's probably a whole lot that we don't know.
Yeah, wow. Well, thank you so much for sharing your work with us. I'm guessing we're going to hear so much more about what you're learning about these incredible bees in Arnhem Land. It's really exciting stuff, Rachel. Thank you. Thank you again for all the questions. I'm so sorry that every time I can't get to all of the fantastic questions that you're asking, but it's just great to see everyone engaging with this incredible science. So we've come to the time of the evening that we are ready to welcome our final pitcher to our stage, Dr. Wing Yan Chan. Wing's been waiting for a long time. We're finally there. So as well as being an amazing scientist like all of the pitchers that you have met this evening, Wing is also a helicopter flight instructor. So when she's not down on the ground here with us doing amazing science, she's up there above the university flying with her pilot students. And tonight, Wing is going to tell us about the secrets of extreme corals. So take it away, Wing. Thank you.
The Great Barrier Reef is a World Heritage. It is our national icon. It belongs to us. As a child, I lived near Tolo Harbour in Hong Kong. In the 1800s, it had a beautiful coral reef with up to 80% of coral cover. But that was history. By the time I was born, less than 1% was left because of the environmental change. I wish I could go back in time to change this, but I couldn't. Yet there is still hope for coral reef across the world. Therefore, my passion is to develop tools to study, protect, and restore coral reef worldwide, and preserving coral reefs for our future generations. To do this, I moved to Australia because I want to learn from one of the best conserved reefs in the world, the Great Barrier Reef. Experts estimated 70 to 90% of today's tropical coral will die in the next 10 to 15 years. My research focuses on hope. Across Australia, some coral thrive in an extremely hot environment, just like the predicted future. Here's a key fact about coral. Coral survival critically depending on the tiny algae that live inside them. This algae transfers nutrients to the coral. High temperature is bad for the coral because it breaks down this partnership. The coral starves and they often die. But this is not the case for extreme coral, and therefore I want to know their secrets. I will unlock the secrets in 3 steps. Step 1. Collaborating with universities and the ecotourism community, I have identified 2 extreme reefs like this in Australia and will collect a small fragment of these corals with their help. Step 2. Using genetic tools, I will identify the algae partner of extreme corals and test whether they can survive and provide energy under high temperature. Step 3, and the most exciting, I will bring the latest imaging technology developed from my early career researcher grant at the university. This new technique will map nutrient exchange between extreme coral and their algae partner, identify what contributes to coral nutrition and resilience under a warming ocean. Discoveries from this project are readily translated into innovative bioengineering solutions. Algae that can support coral nutrition can be grown in the laboratory. They can be carefully given to the coral to form a new partnership with enhanced resilience. Or, compounds that can support coral nutrition can be artificially made. They can be given to the coral as a nutritional supplement to help them to survive through ocean warming. My mission is to find hope for Australia's coral reef and ensuring our future generation will not experience what I did as a child in Hong Kong. We have no time to lose, and I need your support.
Wing, thank you. And I'm sure everyone at home is also hoping that you don't have that same experience again of losing our coral reefs. Lots of questions. The first one I want to ask is just for people who aren't coral reef biologists. What benefits do the algae get from this relationship with the coral? Presumably if it's a partnership, it's a two-way street.
It is a two-way partnership. So for the algae, they get two things. The first, they get protection. They are in a safe environment, protected from the coral. And the second thing, they also get nutrients from the coral as well through their waste product.
Right. Okay, excellent. And what nutrients do the algae actually provide for the corals? Like, what do corals like to eat?
That's a good question. So some of the key things we know are sugar. So things like glucose are one of the key products we know are provided from the algae to the coral. There are also other chemical compounds, such as amino acid and fatty acid, but there are a whole suite of compounds that are provided from the algae to the coral. Some of them we don't even know, we don't even identify that just yet.
That's why you've got to be out there working. And how different are the coral species around, in different parts of the world? The work that you're doing here, I know you've got a lot of partnerships in different parts of the world, but is the work you're doing here, will that have relevance for other parts of the world as well?
Yeah, absolutely. So the reason why we want to study this extreme coral is we try to understand two things. So the algae partner themselves and also the compounds that are providing nutrients to the coral. So even though we're studying Australian coral reef, of course, but this algae partner also can find in different parts of the world as well. In other words, if we can find those right partners that can provide nutrients to the coral under a future condition, they can be potentially used in other parts of the world as well. Mm-hmm. And the same for the compounds that are benefit for nutrition for the coral. They can just be used as a nutritional supplement.
Mm-hmm. Fantastic. Ali's got a question saying, interesting that we study fungal-algal relationships, and now you're talking about algal-coral relationships. Can you tell us more about relationships that algae find themselves in, I suppose? And a question about, is this project useful for obtaining secondary metabolites?
So the first question is about what kind of other partnership these algae also form. So they do form partnership with other marine species, for example, giant clams. They are known to form partnership with, but it's different species. So there are 15 genera that we know from at this moment of time for these algae species. And in addition to that, other things like jellyfish, and so it's quite a suite of marine animals that they form the partnership with as well. The second question is about secondary metabolite. I'm not sure exactly what he or she is trying to get.
No, I'm not entirely sure with that one either. Ali, if you want to give us any more information, let us know. Maybe I'll go on to Peter's question, and that is, will corals be able to form a relationship with different species of algae if they are found to be more tolerant of warmer temperatures?
Yeah, that is a really good question. I'm so glad you asked it. So
Thank you, Peter.
One of the key area of our team in research is to look at finding this resilient algae, whether it's naturally occurring in the reef that we find them or developed in the lab by us. And so one of the papers going to come out tomorrow, so watch this space from our lab, that we demonstrate we are actually able to change the algae partner of the coral and then help them with resilience. So watch this space for tomorrow.
Ooh, that sounds very exciting. You heard it here first, everybody. Robert would like to know whether there are bacteria that work with the symbiotic algae in the corals.
Yeah, absolutely. So corals are really fascinating, complex animals. So they have partnership with algae, but also with bacteria, archaea, fungi, anything else. The bacteria live inside some of the algae partner, and some of them live outside in the algae in the coral as well. So different algae partner are known to have some different bacteria. Whether or not how they interact with each other is another area to study.
So there's so many things that you need to keep working on, Wing, so we can find out more. The next question is, if there's only a few coral species we know of that can survive at high temperatures, what are we talking about in terms of big picture impacts on reef ecosystems when we think about climate change?
Yeah, I love this question. So in extreme environment, it's actually not true that it's not always that we found a reduction of species diversity. This is often true, but in some cases, it's actually to have just equally as diverse in extreme environment. And that's really interesting because some of these species, typically we anticipate them to be vulnerable to climate change. However, they actually do thrive in this extreme environment with high temperature or quite acidic environment. So we don't understand why, and therefore this project basically seeks to understand why they are resilient and then using their secret to help other corals to survive through ocean warming.
Well, I think we all hope that you can find out their secrets. I guess what I'm most interested to find out, Wing, is are you feeling hopeful? I mean, we hear so much bad news about our coral reefs and about how our natural world is being impacted by climate change. You're obviously doing this incredibly important, exciting work. Do you feel hopeful?
I'm absolutely hopeful, and we need to have hope. However, there's a catch. So hope cannot just be hope, and it needs to come with an action. So my favorite definition of hope comes from Dr. Jane Goodall. Many of you have heard about Jane, her work as a primatologist, but also empowering young people and community to make a difference in chimpanzee conservation. In her recent book, she defined hope as something we desire to happen, but we need to be prepared to work for it. So in other words, we can't say we're hopeful for climate change without doing anything about climate change. We need to do something about reducing our carbon footprint. So I invite all of you to have hope. Remembering hope comes with action, and together we will be the hope for our future generations.
Oh, Wing, it's such a wonderful way to end our pictures this evening. And I think we could all stand to learn a lot from the incredible Jane Goodall. So thank you for sharing that with us. Remarkably, we are now at the end of our pitch presentations. I hope you have been as enthralled as I have been learning about this incredible research that our scientists are doing. Such a diversity of amazing topics and this incredible call to action of all of the remarkable things that we are currently finding out. It's such a privilege for me to have the opportunity to speak with our incredible pitchers. And the fact that we have now heard from each of our speakers means that it is time for you to cast your vote in the People's Choice Award. The award is now open. So please make sure you have your say, cast your vote. The winner of the People's Choice Award this evening will receive an additional $10,000 in research funding. That's separate to all of the other prizes and separate to the $10,000 that each of our pitchers has already been awarded to help them with their research. So it's time to have one last word from our judges before they have the incredibly difficult task of picking our top 3 pitchers for this evening. As I've said, I'm glad it's them and not me. So Mick, I don't envy you.
Thanks, Jen, and thanks to all our pitchers. There's just so much amazing talent in the room tonight, and we've got a really difficult task ahead of us, but I think you'll agree that they've all presented thorough and exciting research projects that are going to have lasting impact. As Deputy Dean, I'm really proud to see such fresh ideas coming from the Faculty of Science and to be able to lend our support to these emerging researchers. The future of science is bright. Just before the judges head off to deliberate, I would also like to introduce Dr. Ellen Cottingham. As I mentioned earlier, Ellen won the Big Science pitch last year. So I'd like to hand it over to Ellen. Can you tell us about your experience in the pitch and what it's done for your research?
Absolutely. So I won the Big Science pitch last year. I entered with a pitch where I was trying to come up with a new way to get rid of one of Australia's newest invasive species, which is a little salamander. It's called a smooth newt, and it arrived in Melbourne in 2011 and has since spread, unfortunately, and it very much poses a risk of being the cane toad of the south, as we call it. And so it's a really difficult to manage species, fundamentally because we don't know much about it. So with the Big Science pitch money, we were actually able to sequence its genome for the first time ever. Anyone who knows anything about amphibian genomes, I didn't before this project, they're very, very hard to sequence. So that was completed very recently, and we were also able to purchase the reagents and equipment to actually make the first smooth newt that carries in its DNA a way to basically spread its infertility through its comrades and essentially breed smooth newts out of extinction in Melbourne. And so the Big Science pitch has been absolutely, just so incredibly helpful at progressing this important work. So I'm very grateful to be one of the Big Science pitch awardees. So thank you, and back to you, Mick.
Thanks, Ellen. Really great to hear about the work and how it's advanced, and I'm sure you'll all agree it's been great to have you along to share that with us, so thank you very much. It's now time for the judges to go and deliberate, and so I'll hand back to Jen.
Thank you, Mick. Good luck. Have fun. I hope it doesn't come to fisticuffs as you try and make a decision. So we do need to give our judges a few minutes. And so what better to do with our time than have a little bit of trivia to test just how well you've been listening to our pitches this evening. So our online team is going to share some trivia questions with you based on some of tonight's pitches. You can see our first question is already there for you. So get your thinking caps on. The first question comes from Wing's work. We thought we'd make it easy by starting with the most recent pitch that you've just heard. And you can see the question from Wing is, why are algal symbionts important to a coral? Is it that they defend the coral from predators? Is it that they provide the coral with nutrients? Is it that they protect the coral from excessive sunlight? Or that they provide the surface for a coral to grow on? So I'm only going to give you another moment before we share the answer. I'm wondering how long you need to remember what Wing was talking about and to remember all about algal symbionts. I'm thinking that that's probably long enough. Let's see if you got the answer correct. You'll remember that Wing told us that algal symbionts are important to a coral because B, the second answer, they provide the coral with nutrients. And Wing answered for us exactly what those nutrients are. So well done to you if you got that one correct. Our second question is from Brendan. And Brendan wants you to remember his conversation about shellfish reefs. And he asks you, are shellfish reefs important habitat for A, crocodiles, B, jellyfish, C, fish, or D, drop bears? Now, I'm thinking Brendan is having a little bit of a lend of us here. I'm not sure how many international guests we have joining us this evening who are thinking, what on earth is a drop bear? Brendan, you might have to fill us in later for anyone who is wondering. Is it crocodiles, jellyfish, fish, or drop bears? You won't be surprised to hear that the correct answer is fish. No drop bears in our shellfish reefs. Thank you, Brendan. All right, our next question we have for you is about Rachel's work. And Rachel would like to know, how much honey does a hive of the common sugarbag bee species, which is Tetragonula carbonaria, let's hope I've pronounced that correctly. How much honey does a hive of the common sugarbag bee species normally produce per year? Is it enough to fill one 500ml jar? Is it 5 jars, 10 jars, or 100 jars? Now I'm thinking how small these bees are. I'm thinking 100 jars would be quite remarkable. Time for you to cast your vote. And if you said that you thought it was enough to fill one 500ml jar, then you are absolutely correct. I think these little bees would have a lot of trouble coming up with 100 jars worth of honey. All right, let's give our judges a little bit more time. It is time to think about fairy wrens. How does Claire suspect that the harsh weather is affecting superb fairy wren egg production? Does she suspect that what we're going to see is larger egg sizes, or smaller egg sizes, or larger clutch sizes, where you'll remember the clutch is how many eggs in the clutch, or a smaller clutch size? So is Claire expecting for the size of the egg to change, or the size of the clutch to change? Think back to Claire's fantastic pitch. And if you chose smaller clutch size as the correct answer, then you are absolutely right. All right, let's move on to our fifth question. And this question is from Josh. Which answer does not relate to bold fish? So, bold fish are often more aggressive, or bold fish require more food, or bold fish are more competitive, or bold fish are naive to predators. can you remember what Josh told us about his bold fish? Is it more food, more aggressive, more competitive, or naive to predators? If you selected that bold fish require more food, is the correct response, because in fact it's the incorrect response. We know that bold fish don't require more food, or at least not that we know about. Then you are correct. All right, I have been given the excellent news that our judges have reached their decisions. I hope you enjoyed our little trivia quiz. I'm sure you did exceptionally well, and I'm pretty sure no one will have guessed drop bears, although if you did, that's fine too. Nothing like a good bit of trivia to keep us going. But our judges are ready to announce our top 3 pictures. So Mick and team, just please put us out of our misery. Who has taken the top honours this evening?
Well, the panel has conferred, and it's been a lively and robust discussion. I'm pleased to say there were no fisticuffs, but it's been a very difficult choice to pick the winners from a fantastic array of presentations and really great science, and all would be very worthy of funding. In awarding the prizes, we looked for a combination of innovation and in the approach, the possible impact of the work, and the delivery as well. Tonight's prizes were made possible thanks to the generous donations to the Native Australian Animals Trust and through the Robert Johansson and Anne Swan Fund. Without further ado, I'm going to pass to Ellen and then Tanya and Christian to announce the prizes. So first to you, Ellen.
Absolutely. So I'm happy to announce the 3rd place winner, which is Win Yan Chang. Congratulations.
Over to me. So it's my great pleasure to announce the 2nd place winner, which is Rachele Wilson. Congratulations.
And it's such a big privilege to announce the winner tonight is Dr. Laura Bibiana Ospina-Rozzo. Massive congrats.
It's been a fantastic time hearing from the pitchers. Congratulations to all of them. We've had a really enjoyable time on the judging panel, and I hope you've had an enjoyable time watching as well. There's a huge amount of talent and passion and acumen in this room, and I'd like to thank you all. I'd like to thank our judging panel for deliberations. Thank you for taking the time and joining us in what proved to be a very difficult task and judging the calibre of the presenters this evening. For our audience, I hope you've enjoyed hearing about the 6 new research projects of our early career researchers. Each pitch has shown what can be achieved when we focus on Australia's native animals and their environments. I hand back to Jen for the announcement of the Winton's Choice and the People's Choice Awards.
So it's my great pleasure to announce the winner of the Tim Winton Award. Tim Winton is the patron of the Native Australian Animals Trust. Sadly, Tim isn't able to be with us live this evening, but he did watch all of the pitch videos earlier in the week and sent me a message to read out to you on his behalf. So these are words from Tim Winton. Tim says, "First, let me say that these are all terrific projects, and this year they've been exceptionally well pitched. Every one of these endeavours addresses an urgent, real-life issue that's a result of anthropogenic pressures. The widespread loss of temperate oyster reefs, the fragmentation of those rivers that we haven't yet destroyed entirely, the likely loss of most clearwater corals, and the loss in abundance and diversity of native birds and insects because of our disordered climate. Congratulations to all these scholars. There's so much promise here and so many reasons to support these projects. Honestly, if I had my way, I'd support all of them, but I am only allowed to choose one. I'm a big believer in two-way thinking and two-way science, and I'm attracted to research with direct implications, culturally and materially, for the most vulnerable communities who are trying to survive in the Anthropocene. So this year, my vote goes to Rachele Wilson for her work on sugarbag bees in collaboration with the Yungu people in Arnhem Land. Massive congratulations, Rachele. Wow, I'm very excited that we've heard so many exciting announcements already, but of course there is still one to come, probably the one that you are most excited about, and that's because it's the one that you got to vote for. I've just been told who the winner of our People's Choice Award is. It's always the most exciting because we can't predict what's going to happen on the night, and it was a very close race, as you might predict, with only a few votes between our top two favourites. But I'm really pleased to say that the winner of the 2023 Big Science Pitch People's Choice Award is, drum roll everybody, drum roll, is Rachele Wilson. Well done, Rachele. But wait, there is more. I'm really excited to say that I have very up-to-date news and a special surprise announcement that before we conclude our evening, we actually have one more award that our pitchers don't know anything about yet. This is coming as a surprise. So for the first time, it's my great pleasure to award an additional prize of $10,000, which has been generously donated by the School of Biosciences here at the University of Melbourne. And this award has been judged by three of our school's most eminent scholars, Professor Lynn Selwood, Professor Mick Keogh, and Emeritus Professor Roger Cousins. And they've chosen the project that they feel shows the most innovation and impact. And I'm really pleased to announce that the inaugural winner of the School of Biosciences Big Science Pitch Award is Dr. Win Chan for her project on the secrets of extreme coral. So what's left to say is, of course, congratulations to all of our pitchers. I'm sure you'll agree with me, they are doing extraordinary and important work, and it's such a pleasure that we are able to support all of them in the work that they're doing. So I get to do some thank yous. Thank you, of course, to our judging panel, to Mick, Ellen, Tanya and Christian for thinking so much about how you would best like to award these funds generously donated to the Native Australian Animals Trust. Difficult decisions, but excellent decisions that you've made. I'd like to thank all of the science faculty event team and all of the talented crew that are in the studio with us this evening. An event like this doesn't happen without a lot of really skilled expert people doing their job superbly. So thank you to everyone, with special thanks going to Amber and Lee. Of course, a huge thank you to the Native Australian Animals Trust for the very generous prize money and for supporting our up-and-coming research superstars in such a meaningful and practical way. Thank you to our pitchers, of course. Without them, we don't have such an exciting event. In fact, we don't really have an event at all. So thank you to Laura, to Brendan, to Claire, to Josh, to Rachele and to Wing, who I think you'll agree have done an extraordinary job this evening telling us about their work so clearly, so succinctly. 3 minutes is not very long after all. Finally, a huge thank you to everyone who has joined us online to support our pitches. Thank you to you for coming, for setting aside the time this evening to be with us, for asking such fantastic questions, and for casting your vote in the People's Choice Award. So congratulations, everybody. Thank you so much. It's been a really wonderful evening, I'm sure you'll agree. And I really look forward to seeing you next year for our 2024 Big Science Pitch. Thank you again and enjoy the rest of your evening.